Chandra from Chittaranjan National Cancer Institute, Kolkata, India. And ma'am will be talking about Cobalt Schiff base complex, a ray of hope in the arena of breast cancer therapy.
At the outset, I would like to thank the organizer for inviting me here. I am Dr. Arpita Chandra from Chittaranjan National Cancer Institute, Kolkata. My topic is Cobalt Ship-based complex, a ray of hope in the arena of breast cancer therapy. The worldwide breast cancer is on rise. And the same scenario is observed in India also. The worldwide incidence of breast cancer is about 24% and mortality rate is 15%. Whereas in India, the incident rate and mortality rate is 26% and 22% respectively. Different therapeutic modalities are available to address breast cancer. Our focus is on chemotherapy. More specifically, the metal-based chemotherapy. After discovery of cisplatin, the metal-based chemotherapy get a tremendous impetus. So, with this metal-based chemotherapeutic drugs suffers from severe toxicity and resistance or cancer relapse phenomena. So researchers tried to develop the second generation or third generation anti-cancer drug by keeping the metal center fixed and changing the surrounding ligands. In such scenario, oxaliplatin, a platinum-based anti-cancer drug, came into picture, bearing diamino cyclohexane ligand. This oxaliplatin, however, overcome the cisplatin resistance, it is also have systemic toxicity, have suffered from resistance and also cancer relapse. So researchers are thinking in a way that we are focusing on the ligand system and keeping the metal center fixed. If we change the metal center, then toxicity due to heavy metal may be reduced. So, in quest of non-metal platinum drug, researchers move forward and they uh, did extensive research, and we found that the particular sheep-based metal complexes have a positive role in as a cytotoxic agent. And in our review we published in Polyhedron, we found that sheep-based metal complexes acts against many type of cancer. There are different type of sheep-based cobalt complexes. Some are mononuclear, some are dinuclear, and we find that mononuclear complexes are mostly effective against various type of cancer. During the review procedure, we found that the mononuclear cobalt bearing ship-based complexes mostly effective against breast cancer followed by lung cancer. So keeping this backdrop in mind, we have synthesized a ship-based metal complex. Ship-base is This is a sheep base. This C double bond N is called the imine bond, and this is a aldehyde. Basically, C double bond O is called the carbonyl group. When a carbonyl group is condensed with the primary amine, we get this type of C double bond N. This is a very popular type of ligand in the field of chemistry due to its diverse application of the sheep base itself and also its metal complex. Then we condense this ship-based complex with a cobalt nitrate salt, and we took cobalt because the cobalt is an essential trace element, and if any toxicity arises during the course of treatment due to cobalt, body may overcome this kind of toxicity as the body naturally has cobalt in it. Then we we developed this novel complex and we characterized it through X-ray crystallography and the other physicochemical analysis like IR, CV, UV, mass spectra, and then we purified, and with HPLC study we found that this compound is 99% pure. With the pure compound, we evaluated its biological activity on the breast cancer cell lines. We took two breast cancer cell lines, MCF7 and MDMB231. One is luminal A subtype and another one is TNBC. Firstly, we performed a cytotoxicity study and we found that. May I have that microphone, please? We found that the data show that when we compared with oxaliplatin, it shows cytotoxicity at lesser concentration than oxaliplatin. And we tested it against PBMC cell line and its IC50 value is greater than 60 micromolar. Therefore, we may conclude that the complex is non-toxic in at its IC50 dose. Apoptosis analysis shows that the compound shows late apoptosis like oxaliplatin, and also nuclear fragmentation is observed under microscope when we stain this treated compound treated complex with DAPI staining, and with CFAC proliferation assay study, we found that the complex restricted the proliferation rate, and also found that it arrested the cell cycle at G2M phase. With wound healing assay, we found that the complex restrict the movement of the cancer cells. Further, this study was supported by this Western blot analysis. We found that the G2M cell cycle arrest related proteins are differentially regulated in this case. And also the apoptotic protein, the P53 is and BAX is elevated as expected, and BCL2 and BCLXL, which are anti-apoptotic protein, are down-regulated. This is the mechanism at a glance. Now we are moving towards the MB231 cell line, where we found that it shows the IC50 almost equal to oxaliplatin, and with DAPI staining we observed the nuclear fragmentation. Annexin V PI dual staining showed that oxaliplatin and our complex both shows the late apoptotic rise in late apoptotic population. That was further validated by tunnel assay, and CFAC assay showed that this complex also restrict the proliferation. As the different metal complexes induces apoptosis through intrinsic mechanism, so there is a involvement of mitochondria. So we checked for is there any difference in mitochondrial membrane potential, and we found that depolarized mitochondrial membrane. And due to this depolarization of mitochondrial membrane, there is translocation of cytochrome C from mitochondria to the uh, cytoplasm. This is clearly evident from this Western blot data. And also uh, there is a rise in SMAG diablo in the cytosolic fraction, and decrease in the mitochondrial fraction, and the inhibitor of apoptotic protein survivin is also decreased. This is the Western blot analysis which suggests the PI3K-AKT pathway is activated. Uh, Activated pathway is down-regulated with the treatment with our complex. And complex also arrest MDA-B23 cell line in the G2M phase with further supported by Western blot analysis. This also block the migration as found from the wound healing assay. Also, migration and invasion was restricted due to presence of this complex. This is the mechanism at a glance. This is the animal study we performed on the biopsy mice bearing 41 tumor cell, and we with the toxicity study we found that in subacute and chronic there is no hematological or biochemical toxicity, and in chronic and subacute study we also found there is no body mass alteration of the mouse. And the with the histology study we found the compound impart no significant toxicity on liver and kidney cells. And this is the image under in vivo imaging system where we injected mice with 41, and then IV injection was given in the tail main, and we found a significant reduction in the tumor size. And this mice also, the tumor size also monitored with slide calipers, and we found the significant reduction in tumor volume. And this compound also has a protective, maybe have a protective role against liver and spleen as evident by this picture. And this is the data from the tumor, where we find that the cell proliferation marker Ki67 is significantly reduced after treatment, and also other Western blot analysis suggested that here is the PCNA data that also suggests that the proliferation is reduced. And this is the tissue from the treated and the tumor cell, which shows the significant improvement after treatment. And survivability of the mice also increased. So thank you.